Welcome to Lesson 30, Cleaning Documentation and Cleaning Limits. In this lesson we're going to be looking at documentation like cleaning validation protocols, reports, how to calculate cleaning limits, post-validation activities and revalidation activities. Performing cleaning validation. Documentation includes master validation plan, your validation protocol and your validation report. You might want to be doing your cleaning validation plan in stages. You might have a number of protocols that will come together to give you a final document, or for a smaller, less complex piece of equipment for cleaning, you might just have one. Your cleaning validation protocol should include the following: the objective of the validation process, the responsibilities for performing and approving the validation study, the description of the equipment to be used, the interval between the end of production and the beginning of the cleaning procedures, the cleaning procedures to be used for each product, each manufacturing system, or each piece of equipment, the number of cleaning cycles that need to be performed consecutively to give you a passing result, any routine monitoring requirement, sampling procedures, including the rationale as for why a certain sampling method is used, so where you're going to sample and how you're going to sample, so you should be picking out your worst case areas for those. That's how you clearly define your sample locations. Data on your recovery studies if that's applicable, so have you shown that you can get your residues returned from your equipment. Analytical methods, including the limit of detection and the limit of quantification of those methods. The acceptance criteria, including the rationale that you have for setting the specific cleaning limits that you have. Other products, processes and equipment for which the planned validation would be valid according to your bracketing or your grouping concept. And when revalidation would be required, if in fact it is. Cleaning tags and logbooks. To prevent equipment, component, rooms or process lines, etc. going over their clean hold times or being inadvertently used when unclean, they should have a logbook or a tag to denote their clean status. That is a really important part of cleaning validation documents. Your cleaning validation report then. Your report should cross-reference the cleaning protocol. It should mention the area or the piece of equipment that is to be cleaned. It should also talk about the product that was previously used on the equipment, who performed it, the time that it was cleaned at, what SOPs were used for the cleaning process, any sampling and the test results associated with that sampling, a comparison of your test results with your acceptance criteria, who was it signed off and approved by. The report will hopefully conclude that your cleaning validation was successful. If it wasn't, you would need some more documentation then to discuss why it wasn't and an investigation as to where you go from there. Acceptance limits for cleaning validation. When determining cleaning limits, remember, cleaning is carried out for two reasons: to protect the patient, number one, and number two, to protect the people working with the equipment, so your operators and your maintenance people, etc. Limits are referred to as ARL, so your acceptable residue limit, or you might hear it talked about as MACO or MAC, your maximum acceptable carryover. Acceptance criteria must be set for the following items during cleaning validation: the residues of the active ingredient, the residues of the chemical raw materials and the intermediates, the residues of the cleaning agents themselves, and any microbial residues or endotoxins. The acceptable limits for residues after cleaning, your ARL or your MAC, must be determined prior to cleaning development, analytical method validation, visual limits, and recovery factors. There are different approaches for calculating your cleaning limits. There is a 10 ppm criterion, a dose method, which is also called the therapeutic approach. There's a permitted daily exposure limit, your PDE, and then you've got visually clean, where you have no quantity of residue should be visible on the equipment after cleaning is performed, so it should be visually clean. All criteria really should be met, so what this means in practice is that you must use the most stringent criteria to ascertain your carryover and your acceptable level of residues. The 10 ppm method. This is defined as an allowance of 10 milligrams of hazardous contaminants contained in 1 kg of product A going into subsequent product B. That's equal to a 10 ppm ratio of subsequent product. It may not be the most appropriate method when medicines of variable efficacies are manufactured in the same equipment. The calculation for a 10 ppm method is using your MAC, so your maximum allowed carryover. So you've got your batch size in kg over 10 to the power 6 multiplied by 10. dividing the batch size by 1 million to convert it into ppm, and you're multiplying it then by 10 to convert it into 10 ppm. So this gives your 10 ppm maximum allowable carryover per batch. This has to be adjusted then for the area that is swabbed, so it could be 100 square centimeters, for example, if that's your specified swab area. The swab limit then is calculated by using the MAC per 100 centimeters square, or whatever your adjusted amount is for your swab size. So you've got your MAC of your batch from your calculation above, divided by your product contact surface area, multiplied by 100, and that gives you your 10 ppm method. The dose method then is defined as having no more than 0.01 dose of any product appearing in the maximum daily dose of another product. This method assumes that 1/10 of the usual dose of a pharmaceutical product is considered to have limited to no pharmacological effects. To that, a further safety factor of 1/10 is applied. As this is a theoretical approach to evaluate a real process, an additional factor of 1/10 is used to take variations in the cleaning process into account. So that gives you a calculation, a total factor of 1/1000, maximum allowable carryover from one batch into the next. So your maximum allowable carryover is 0.001 multiplied by your minimum daily dose of your product A, multiplied by the batch size of product B in kgs by 10 to the 6th, all over the maximum tablets per day of product B, multiplied by the tablet weight of product B. This needs to be adjusted for the area that is swabbed again, so you're using your maximum allowable carryover. That's milligrams over 100 centimeters squared, or whatever the adjustment is needed for your process. So your maximum allowable carryover per batch from the calculation above, all over the product contact surface area, multiplied by 100. In the dose method example, product A will be cleaned out of the process stream. The product has a minimum daily dose of 10 milligrams and the batch size is 200 kg. The next product B has a maximum daily dose of 250 milligrams and the batch size is 50 kg. Both A and B are administered orally, and the sensitivity factor is set to 1000. So you're going to calculate the maximum allowable carryover for A and B. Your maximum allowed carryover is your 10 milligrams of your daily dose multiplied by your batch size in milligrams, all over your sensitivity factor multiplied by the maximum daily dose of B. So that gives you 2000 milligrams. Permitted daily exposure, PDE. It is based on the determination of safe threshold levels after a review and evaluation of pharmacological and toxicological data of individual active substances. The procedure for the determination of health-based exposure limits for a residual active substance is based on the method for establishing the permitted daily exposure, PDE, as described in your EMA guide. So have a read of that, it's worth having a look at, and your Appendix 3 of the ICH Q3C Impurities Guideline for residual solvents. So there's a whole science behind this, and it's quite complicated and complex. The PDE represents a substance-specific dose that is unlikely to cause an adverse effect if an individual is exposed at or below this dose every single day for a lifetime. The determination of a PDE involves hazard identification by reviewing all the data, identification of critical effects, determination of the no observed effect level, NOEL, of the findings that are considered to be critical effects, use of several adjustment factors to account for various uncertainties. Your PDE is calculated, again looking at a swab of 100 centimeters squared, so that's your PDE multiplied by your batch size, multiplied by 100 centimeters squared, multiplied by your recovery factor all over your maximum daily dose in milligrams, multiplied by your surface area. Visually clean. We talked a lot about visually clean in our previous lesson, but the basis for this criterion is that there should be no quantity of residue visible on the equipment after cleaning procedures have been performed. A number of products might meet the 10 ppE dose and the PDE criteria at levels where there still could be visible residues left on the equipment after cleaning. If the quantity of safe residues is enough to be still visible, the equipment must still be cleaned until no residues are visible. So that comes into your making sure you're working from the most stringent criteria. The regulators. Inspectors now look for evidence that the cleaning processes are developed to guarantee that they will reduce the critical residues below the health-based exposure limits. Note that the maximum allowable carryover, the MACO, should be justified from the health-based exposure limits and process capability. Post-validation activities. A cleaning process can fail even after validation. Cleaning verification should be performed frequently to confirm the effectiveness of the cleaning procedure. When to revalidate. The first reason is due to changes For example, if you change your cleaning agent or your concentration, if you changed your cleaning procedure, parameters, or the equipment. Some cleaning processes are considered so critical on the site and they will be required to have regular periodic revalidation, and this will be documented in the Site Master Plan or in an SOP. APIs. Many stages of APIs do not have medical doses, so a toxicity approach commonly used to determine your ARL or your MACO. Biopharmaceuticals. Cleaning validation is very expensive and time-consuming. Therefore, the biopharma industry is moving towards single-use equipment. For example, vessel liners, tubing, disposable filling needles, plastic one-use containers, etc. The wave bag for cell culture pictured here will be disposed of after a single use. Steam in place between runs means proteins are deactivated, so there is no carryover. Cleaning validation requirements then are minimal. Single-use technology often means little cleaning validation is necessary. There's a lot of references here for you to have a look at, and particularly do have a look at the EMA guideline on setting your exposure limits and cleaning validation for the 21st century as well. So these will be in your appendix section attached to this lesson. Over to you. What documents do you require for cleaning validation? Identify the methods that can be used to calculate acceptance limits for cleaning validation. Thank you for your time and attention.